Hello, everybody. It's a great pleasure to be with you again. Thank you, Troy, and thanks, Harold, for organizing this webinar. It's a great opportunity for me to discuss our recently published paper. So we are presenting session two of the webinar, which is again on the carbon stability in biochars. Today, I'm going to talk about novel proxies, most importantly, inertinite benchmark that many of you have asked, and how it is being used for evaluating carbon stability and permanence. So before that, I would like to acknowledge EBI, Geological Survey of Denmark and Greenland, GEUS, and of course, Aarhus University, and most importantly, my co-author, Dr. Henrik Ingemann Peterson from GEUS. I would also like to mention there would be another session, session 3, which would be more discussing about industry-related material, particularly the efficiency of carbonization as it relates to permanence, and that's exclusive for EBI members. So I will be discussing primarily our paper, which was published in January, assessing biochars permanence and inertinite benchmark. It is a free access paper. You could Google it, and you'll be able to read the whole paper. And I'd like to acknowledge all my co-authors that were involved in this research. Okay, let's get to it. Biochar permanence experiments. I discussed briefly this in our last session. There are two important methods that are being used for measuring permanence. One is a field trial, and the second one is soil lab incubation. Of course, the lab incubation is the most important one, quantitatively speaking. This is because the degradation of biochar in a soil bacteria is conducted in a closed system, so the exact amount of carbon loss can be measured, and then the mass balance of carbon can be performed. So primarily this method is used for modeling the permanence, particularly because it doesn't have a physical and erosional loss of biochar. So there has been many, over hundreds of studies, 120s, more than 120 studies on the lab incubations and soil incubation of biochar by many authors over the past decade. All of these have been compiled in a great paper by Azzi et al., which just came out in 2024. I invite you to read it. It's a great paper. And especially the metadata associated with the supplementary information is extremely useful, as you could go through the data and search what you're looking for. But basically, I excluded the biochars below 550 degrees centigrade because most of the industry biochars are above this temperature. But regardless, what you could see is a decay of the biochar. So it's a carbon loss shown on the y-axis, and the x-axis. This is time. In this case, most of these studies are in 1 year, 365 days. So, of course, you can see that the amount of loss of carbon during this period is very negligible. It is around 1%, and that's very typical of the biochars that are carbonized at higher temperature. So what is done after is projection of these empirical data 200 years. Basically, see after 100 years if biochar resemble the same decay pattern, how much of the carbon will be lost over 100 years. And important parameters are modeled and suggested. One is mean residence time, which is basically number of years that it takes that an entire carbon in the biochar is being degraded. And the second one, which is more commonly used, is F-perm. This is basically fraction of biochar or fraction of carbon in biochar remaining after 100 years. So this is widely used. So since these incubation studies are of course expensive, slow, it takes years to do that. We can't use these methods for every biochar that's being produced. Therefore, we need some kind of proxies. So we can use these measured proxies to infer the permanence or F-perm. And these proxies are typically are plotted against the known F-perm, and we get the relationship, and this relationship equation is being used. So these proxies are all have one thing in common, and they're all direct or indirect measurements of degree of aromatization and condensation of organic carbon. So methods such as hydrogen to carbon ratio, oxygen to carbon ratios, fixed carbon volatile matter, fused aromatic rings, Raman graphitization, random reflectance, which I talk extensively today, fluorescence spectrometry, and so on and so forth. And most commonly used is the hydrogen to carbon ratio, which has been published by great work by Wolf et al. Against the F-perm, and this relationship has been established, which is being used primarily to infer the F-perm or fraction carbon remaining after 100 years of biochar based on their measured hydrogen to carbon ratio. So the question remains, as you could see, this kind of inferring or conversion is based on the correlation of the line, which we know for many reasons this correlation is never great because there are so many variables. But one other way to look at it is to establish the permanence of biochar, or the point that we know that biochar becomes absolutely permanent. So at what point biochar is completely permanent? This is similar to mineral carbons that we know in CCS or in other methods that is permanent. So the question is there such a thing as biochar being completely permanent? And if there is, what is the benchmark for that? That is when we discussed in the first session of webinar the concept of inertinite. So yes, geology has very strong evidence and years of decades of publications that there is a most stable form of organic carbon in the Earth's crust, which is called inertinite. And for inertinite benchmark, basically we need to look at the chemistry and optical properties of biochar and compare it to the inertinite, which we know it is permanent. And that's one way to see whether or not the biochar or carbon in biochar has reached the level that can be classified or characterized as inertinite. And that would be inertinite benchmark. And one method that is used to measure or at least qualify the biochar as whether or not it has reached inertinite benchmark is random reflectance. And many of you ask the question about random reflectance. So what is random reflectance? Here it is. Basically, in a reflected light microscopy, random reflectance is measured. Essentially, it's a very simple concept, and it's been done for many, many decades, near century. The particles of carbon, or in this case biochar, is mechanically polished to the perfection, and we have a light source, incident light, white light essentially, and the light is reflected on the surface, on the polished surface, and the amount that is bounced back is measured by a photometer or photomultiplier. And these values have been, of course, calibrated universally, and these values define the random reflectance. Under the microscope, of course, this process is being done. And when you see a biochar under the microscope, it looks like this. The reason it's so shiny, the surfaces, it is because these are polished surface of the carbon particles in biochar, and it reflects a lot of light, and it has a very smooth surface. So under the microscope, essentially the biochar is crushed or powderized and mounted in a resin, and the surface is now cut and polished to the perfection. And we see basically these fragments of biochars are like this. Of course, this is just a picture. In reality, there are millions of fragments that are mounted in a resin. And then from the surface that is exposed, random reflectance is measured on many points of carbon, hundreds. It could be done manually on hundreds, 500, or it could be automatically on hundreds of thousands or millions of surfaces. The measurements can be done. And as you could see in this microscopic image, you could see different fragments. Visually, you could see they have a different reflectance. Some are much brighter, as you could see here, and some are darker. So the reflectance, the range of random reflectance on various particles exposed in the sample is being measured. Of course, this is a one single sample. So you could see there are different levels of random reflectance. There's a different range of random reflectance in a single sample. And that, as I explain later, is a very important aspect of random reflectance. But the results that is being produced is basically a random reflectance range, and this is a data frequency. So we will get a histogram which shows a frequency range or distribution of measured random reflectance. So it's a very quantitative measurement, and it is a spatial. It is it shows the heterogeneity of the samples because it has a range, in opposed to a single data measurement. So if we look at just the average of random reflectance in every given sample, we know since the 90s that random reflectance corresponds beautifully to the pyrolysis temperature. So this graph is from Jones 91. And we know for many decades that random reflectance is a function of aromatization of the molecules. So as the molecules of carbon becomes more aromatized and condensed and become more ordered, they would reflect more light. And this relationship is expressed in this equation, which is, you can find it in our paper. So it is a direct measure of the carbonization and subsequent aromatization of organic molecules, okay? We also know that random reflectance, again in our paper we have shown, for over 64 samples that we measure, we know that random reflectance, which is shown in the y-axis, corresponds beautifully to the hydrogen to carbon ratio, which is another proxy for aromatizations and carbonization. So as the carbonizations increases, the hydrogen to carbon ratio decreases. That's what we expect. There is another proxy that we introduce in our paper, is the hydrogen index, which indicates the amount of hydrocarbons in a biochar. It also shows very good correlation with random reflectance. The correlation is even better than hydrogen to carbon ratio, as you could see with the higher R-square values. What's really important about random reflectance, as I said, is because it shows the range. So if this is a random reflectance, here I'm showing you 3 different biochar samples. The first in blue is produced at temperature of 500 degrees. This one, the orange, is produced at 700 degrees. And the gray is produced at 900 degrees centigrade. So you could see the mean value of each sample increases 2.1, 3.9, and 5.3. We know that. I showed you. This relationship is well understood. But the range, the distribution of random reflectance in a single sample is a function of how efficient the heating is, how efficient the recipe of the pyrolysis is. And you could see basically it becomes extremely narrow and efficient as it goes to the highest temperature, 900. That's simply because all the secondary the reactions between carbons and CO2s that would make the carbonization more efficient. But what's really important is a benchmark that we have extensively discussed in our paper, and I mentioned that also in a previous webinar. And that's the number random reflectance 2%. We refer to this number, magic number, as inertinite benchmark. Why do we know that? Again, there is decades of research in geoscience that we know that when organic carbon reach the random reflectance of 2%, they are geochemically and biogeochemically, they are extremely resistant, and they become non-generative, and they become non-degradable. So most of the organic carbon that survive the entire sedimentary process, that means they go to a geological stage, which we call it metagenesis, they exceed 2%. So now you could see this distribution of reflectance becomes very useful, because now we can define out of these three biochars, we see that two of them are well into the range of inertinite, which is at the geological time scale considered permanent. But the first one, which is produced at lower pyrolysis temperature, shows basically part of it is below the inertinite benchmark, and part of it is above the inertinite benchmark. So this provides a mean of quantification for us, which I can show you here. I basically brought the same peak here, same distributions. And now you could see again, inertinite benchmark at 2%. Now these peaks, these small peaks, can be deconvoluted. And by deconvolution, we could see what's the share of the inertinite material in that single biochar, and what fraction is not inertinite. So when we look at this, the peak D and E are primarily inertinite, and they add up to over 85% of the carbon in this biochar is considered inertinite, and 15% of it as a whole is considered not inertinite. So that produces a very reliable quantification of the nature of the organic carbon within a biochar, which directly infers the permanence. So let me open up this discussion a little bit more here. So the inertinite fraction can be seen in these samples of rice straw biochar. It has a mean reflectance of 2.18. And if you see this distribution, this is after a 500-point measurements of random reflector. So anything about 2%, we refer to them as inertinite fractions. And you could see in these samples, the measurements are done here at these spots, 2.57, 2.64. These are the fragments that are regarded as inertinite. They have reached the highest degree of carbonizations. But anything below 2%, there are different fractions here. One is basically the range, something around 1.2% to 2%, so it's below 2%. these we refer to them as semi-inertinite, and it's not it is actually well published. It's a decade-long science and nomenclatures that has been well defined in organic geochemistry. So these fractions of carbon and this degree of carbonizations in the biochar is referred to semi-inertinite. So you could see in the same samples, some of the surfaces are measured at the lower random reflectance, in this case 1.2% as an example you could see. Again, same distribution histogram, and now you see much lower end of the frequency distribution histogram, and these much lower number below 1.2, you could see them in a white light. They are much darker here. And then if we switch it to the fluorescence light, which shows much more reactive forms of organic carbon, you could see they emit very faded yellow-brown fluorescence properties. These are again very well known for us, and it's called liptinite fraction. This is a kind of organic carbon which is more lipid rich, and it is more enriched in hydrocarbons and functional groups, which has a potential to be degraded. So basically, if you subject this sample to the incubation, one could imagine that the bacterial degradation starts from the most degradable fraction, the most labile fraction started from lowest reflectance, and it would go further if we have a geological time scale to demonstrate. So let's explain the liptinite fractions. What is the quantity of that? So we actually measured these liptinite hydrocarbons in over 64 samples of biochars, the industry biochars, and you could see primarily they have a very low liptinite hydrocarbons. These would result in a hydrogen index of below 1. This line is a threshold of 1. And then we have these ones that the hydrogen index is from 1 to 10, and some they are over 10, but these are exceptions, and they're almost always related to the temperature of pyrolysis and degree of carbonization. So these are the biochars that have the lowest quantity of liptinite hydrocarbons and highest degree of carbonization. There is another fraction of organic carbon in a biochars, which you don't even see them in a reflectance measurements. So we would only be able to see them under a fluorescence light. So you could see the vacuoles of this biochar is filled with this blue fluorescing material. So you could see it closer. And these are condensates. These are the free hydrocarbons that are trapped within the vacuoles of the biochar. They're extremely degradable. And we could also measure these, but they're usually very low quantity. You could see these are the entire biochars from the industry, 64 of them. So the amount of free hydrocarbons, and it is measured in a unit of milligram hydrocarbon per gram of char. So it's basically most of them are below 1 milligram hydrocarbon per gram of char. This is a very low quantity, but if this sample is exposed to the incubation, even though if it is entirely composed of inertinite, but has a little bit of this secondary condensate trapped within the cell structure, it still shows a small fraction of biodegradation in a short period of time. But then once it reaches the inertinite, the biodegradation either stops or slow down in a very geological time scale. So we demonstrated in our study of, again, over 64 biochars, the ranges of random reflectance in all the samples. And you could see these are the biochars and the lines are the range. And you could see most of the biochars, particularly in this zone, where they have a hydrogen index below 1 and IBR2 benchmark or inertinite benchmark reflectance values, which is above 2%. So they are considered uh, extremely permanent biochar. So most of them are permanent. And even these ones, some of them, you could see the range goes below the benchmark of inertinite, but some of them are still above the benchmark. So we have shown that over 76% of all biochars that we measured, the industry biochars, are essentially over the inertinite benchmark entirely and hence considered pure inertinite. I end the presentation with the analogy that that I last showed you, and that was the concrete ball, peanut butter, and jam analogy. So the concrete ball is the inertinite, and peanut butter and jam are the more degradable fractions that comes with the biochar, especially if they are not purely carbonized. So with the increased degree of carbonization and random reflectance, so most of these fractions, except some condensates maybe, will be removed and will be purely uh, 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 inertinitized, and that is the biochar above the inertinite benchmark of 2%, which could be regarded or referred to as a certified inertinite biochar. So if these samples are exposed to the bacterial degradation, as you could imagine, some of the more labile fractions start to be degraded, but ultimately is inertinite fraction that would remain permanent, and that's the fraction that we don't have empirical data on their permanence, because simply the bacterial degradation within the time frame that we do the experiments are not capable of reaching degradation of this fraction. So our projection is mostly based on the peanut butter and jam fraction, and assuming that the whole thing is peanut butter and jam. So so what's really important is to use geological empirical data, which is based on vast number, tens of thousands, maybe hundreds of thousands of sedimentary record and sedimentary archives that we know their geological age and their condition of deposition environment, and we have random reflectance for most of them, and we know at what stage and how the carbon survives, and these random reflectance can be used to infer the permanence for biochar. I'd like to thank all the co-authors and students, and of course, again, EBI, Aarhus University, and GIUS. And if there is any question, I'll be happy to answer.
Great. Thank you again so much, Hamed, for another wonderful presentation. I think the peanut butter and jam analogy is so on point, and I'm glad that you're able to demonstrate that in such a clear way. Harald, I know you have some points to get to, but I'd like to just leave it open for everyone with their questions. Please direct your questions to the Q&A, and we'll get to all the questions we can with the time we have.
Yeah, thanks, Troy. And thanks for keeping an eye on the Q&A, because I didn't do so until now. I will try to get behind it. Hamed, oh yeah, now you're sharp again. Yeah, the focus was on on the background. So thanks a lot for this great presentation, and I think now we have a clear understanding about what random reflectance is and what it's capable of doing. So you've shown us actual images of the different carbon fractions. I think this is kind of the first time for many people here in this room. You showed us the inertinite, how it looks, how it reflects, the elliptinite, the free hydrocarbons, how you measure them. So this is basically, you have the means of accurately measuring their presence and their individual amounts. Plus, you have demonstrated in your paper how inert, respectively permanent, they actually are. So for me and for many others, is the question would arise: doesn't this just represent the basis for a new carbon removal accounting for biochar carbon removals? And what is still missing here to get, I don't know, to the implementation? And maybe a prior question, because I don't know if that became clear. How long is permanent? If you talk about the permanent parts of it, what time frames? I mean, you said relevant time frames, but maybe you could somehow quantify that, even though I know you're generally reluctant for doing so.
It is important to remember that we've done a kinetic study, but based on thermal oxidation. Okay, and it shows it goes well beyond 10,000. It is a geological time scale, but I'm sure colleagues in the soil community have a very valid point that soil and biological systems may act different. Thermal oxidation may not be a direct representation of the biological system, and that's a fair point. Then what we know is we have to refer to the empirical data. So if we had thousands of years to do the soil incubation studies, we would probably know better. But we don't. So we only have geological empirical data. And when we see the geological empirical data, we see the inertinite is the primary mode of storing carbon by Earth. That's how organic carbon is being stored for long-term period. That means inertinite is a way, formation of inertinite is a way for Earth to convert organic carbon in such a shape or such a form that is not available for the processes that are typically cycling carbon in the biosphere. So again, we don't have empirical data exactly to tell you how many years, but we rely on the geological records, and that tells you this is the fraction of carbon that is meant to be geologically stored.
Thanks. And so the pieces that are still missing to come to a certification, maybe it's also on someone else to answer, maybe rather on the people who are actually working on certification schemes, right?
Yeah, that's not my part.
Yeah, I would have wished you can just answer everything, but we'll have a separate session on that. Troy, do you want to proceed with the questions, or should I just pick one?
No, I've got one here ready from Dominique Hélène. They write, you showed that RO is clearly related to the temperature level. Is this also true regarding the time of exposure at such temperature? In another word, a same RO distribution, could it be obtained with different time exposure, higher? They write higher / temperature, lower combination, and vice versa.
Yeah, I certainly hope you attend our next session. These are exactly related to the recipe, pyrolysis recipe. But quickly, the first order of influence would be the temperature. The second order would be the residence time. And then there are other factors, but number one is always the maximum temperature. Definitely the residence time of the heat would affect the how homogenized the carbonization is. But the absolute degree of carbonization or random reflectance, the highest point of random reflectance, will be achieved by temperature. So it is directly related to that. But there are sweet spots that that we could discuss that in the next session, and I hope you attend that.
Yeah, Dominic is a member, so I suppose he will be there in the next session.
The next question is a little more general and kind of gets to Harald, what you were talking about within the biochar space specifically. But how much of these insights on biochar is used today in commercial CDR vetting?
That's for Harald.
Well, we didn't exactly, or nobody actually measured it for most of the carbon removals that have been sold. So a large part of those biochars have not been analyzed. We've just facilitated as EBI the sending of samples from different members to the University of Hamed. That means we have kind of a representative sampling, but it's not like in a scientific way or so. So this is something that would still need to be conducted, actually measuring the composition of biochars and determining the differently inert fractions in order to certify the carbon removals. But there's no scheme yet that actually includes this.
Maybe I add something. Please remember, our paper is published in January, so it's very new, and in science it takes time for people to read and comprehend. And the whole thing really came a year ago or less than a year ago, because the science of random reflectance and all of those that you heard, well established in organic geochemistry and organic petrology in geoscience, basically. It's near a century old science. And that's, I met Harald, and then I realized that there is a significant knowledge gap that is very well understood in geoscience. And then we started this collaboration on the set of 64 samples. And these samples are from every feedstock and every different temperature. We did not choose which one to analyze and which one not. It's just all of it. So the publication is highly scientific, of course, but we need to do more. And right now we are actually helping a lot of colleagues in industry and measuring slowly, but it has to be kind of upscaled and make it faster. But it also takes time for the policymakers to adopt it. So we have presented, but more papers coming, and then hopefully the case will be made to satisfy the regulators and other colleagues in the scientific community.
Great. The next question kind of gets down this line, Hamed, and on my end, it comes up as an anonymous user, so apologies, but they thank you for your talk and want to know the differences between the characteristics of biochar derived from algal or cyanobacterial biomass versus biochar produced from lignocellulosic biomass.
So what's the difference?
Yes, if there are any key differences.
Oh, there are a lot of key differences, but once they become inertinite, they're both inertinite. So it basically, the difference is in the material that they generate before they become inertinite. So the algal feedstock, or even sludge, or they have much more lipids, so they produce a lot more hydrocarbons or syngas and oil and all of those. But woody material produce a lot less. So, but once they become inertinite, they're both inertinite, both carbonized matter, and it really makes very little difference, except in the morphology that you could see them very clearly. So, and also in the recipe, and those they definitely make difference, and that's, I'm pretty sure the operators would know that very well.
Great, thank you. The next question comes from Hans-Jörg Lechemüller, and he asks, if you look into a pore in the optical analysis, the optical impression changes. In brackets, he writes, should it become darker? And how does this influence the RO result?
And we never measure RO inside the pore. We never do that. RO is only measured on the actual mass of carbon, on the actual surface of the carbon that is perfectly polished. That means even if you visually see a slight scratch on the surface, the measurements would not be conducted on that scratch. So it has to be perfectly polished on the horizontal plane and exposed to the microscope before you make the measurements. So what you saw in the pores, those free hydrocarbons and those ones, those are not measured on the they're not measured unless we are assured that there is a solid material. Like free hydrocarbons is not a solid material. It doesn't even give a reflectance. It always gives a reflectance of the mounted resin background, which is below 0.2. So and we usually discard those numbers. So it's basically not a measurable values, and it would not be inside the data distribution.
Great. Next question here. What is the effort required, time/cost, to measure the RO and get the frequency distribution to obtain a reliable mean and SD values?
Right now, you know, what the proxies that we're using now, hydrogen to carbon ratio, you still needs a lab and some effort, some substantial effort to measure those values too. And I think we should continue using those methods because they are important proxies for incubations and all of this. But random reflectance, it's very rapidly becoming more accessible. So I can tell you basically the actual act of measurements takes around 30 minutes. So by operator, but it's now becoming automated. So we're trying to make it very fast and automatic. The sample preparation involves some efforts also making a resin polish blocks that we are trying to make that also very fast. So a slightly more effort than hydrogen to carbon ratio, but it's still quite accessible. So it has been used by coal industry for decades and continue to be used. So it is actually a commercial method as we speak for other industries. But for biochar, we are working on that to make it tailored for biochar and particularly permanence measurements. And now we're doing it for many people, and it's getting faster. And as it gets faster, the cost goes down, and it becomes much more available. So it's not some unique, unusual measurements that only professors do in their lab. No, it's highly commercial method, widely used by many people. But for biochar, the way we do it has not been used in this fashion. And that requires a little bit adaptation that we're trying to do it very fast.
Great. Thanks again. We have a question which I think stems more to the discussion from last week, but I think it's really important that everyone has a clear understanding from this perspective. Andrew McLeod writes, it sounds like we are confident about inertinite stability deep underground. Do we have any understanding of this stability in surface environments, such as agricultural soils exposed to sun, rain, chemicals, and regular physical manipulation?
Yeah, that's a fair question. Do we have empirical data? No, we don't. if we had 1,000 years to measure, we did. Remember that peanut butter and jam on the concrete or inside the concrete. If you subject it to the incubation, within a few years, decades, we now have over hundreds of studies. They always give you a couple % of carbon loss, which is quite low, right? And we don't seem to be able to accelerate this reaction to degrade, let's say, 50% of the carbon in biochar. So therefore, what we usually see, we see the decay pattern or decay rate of the most labile or the most reactive fraction, in this case, jam and peanut butter. That's going to be our empirical data. So any modeling or projection assumes that the entire material is peanut butter and jam. Okay? So now you're telling me, do I have some kind of exact information that if I leave the concrete ball or the inertinite subjected to the bacterial activities, how long does it take? No, I don't. But we have used very harsh oxygenation or oxidation kinetic. That means in a pure oxygen, we try to degrade the biochar at a much higher heat. Again, I know it's not exactly the same as what you are referring to, but we try to basically measure how long does it take to break these carbons, the carbon bonds, with a very aggressive method. And we got as expected, we got the geological time scale. It's well above 10,000 years. But in the absence of empirical data for long-term biological activities, we have no choice but refer to the basically empirical data from the geological records. I just turned it around. How do you know in a CCS, for instance, how do people know that when they inject the CO2 under the ground, it will stay permanently? It is basically geological perspective that they use. And that's the geological perspective. They say, okay, if these geological reservoirs weren't permanent container, then all the fluids inside them, such as gas and hydrocarbon, would have come to the surface and would have leaked because they are buoyant and they will come out. Since they haven't, and these fluids have been sitting down there for millions of years, then it is safe to say it is a permanent container of carbon. So same things, we are using geological perspective, and I really don't have any other answer for you.
Great, thanks again. We have another question here that is asking, considering that each RO measurement is using very tiny pieces of biochar, how many measurements should be performed to get an accurate representation on the analysis of any biochar?
Well, we use one biochar sample, like we get it in a bag, and then few grams basically we get, and mix it very well and get a representative sample from that mix. And near a 1 gram or half a gram, and mount it in a resin. That amount contains millions of fragments, small fragments that will be exposed for random reflectance. And automatically we scan the entire sample. That means it measures millions of pixels of random reflectance. That's what we do. But if your question is that few grams of sample is not representative of the biochar product as a whole, then that's a classic case of any kind of analytical measurements. Like hydrogen to carbon would be the same. You could only put like a gram of sample in the machine. So how many biochar sample do you have to use to make a representation of the product? That's a protocol that should be established in maybe some kind of a project, a smaller scale projects, try to do different sample batch and see if the variability is within the error or not. And in that case, you would know if it is if it is representative or not. So sample representation is a protocol that should be established for any measurements, regardless random reflectance, hydrogen to carbon ratio. And I'm pretty sure that's already established by very well reputable organizations like Pure Errors and other. I'm pretty sure they already established. But once you have a representative sample, the random reflectance is measured on a gram of sample or half a gram, and we basically scan the whole thing.
Great. Next question is from David from EcoEra, and he's wondering if it would even make sense to monitor inertinite carbon over time, considering if it's going back into a soil ecosystem or the biosphere, just based on the inertinite properties of the biochar.
Look, we have over 15 million gigaton of organic carbon in the sedimentary basins. So how do they end up going into the sedimentary rocks? They're all going from the surface. All the biomass is once produced on the surface. And then they gradually either become inertinite, or due to rapid carbonization or wildfire, they're basically sitting on the soil or sediments or on the lakes or oceans, and then gradually they will get degraded. They will get deposited and precipitated, and they go through 3 stages: diagenesis, catagenesis, and metagenesis. We know all of these as a geologist. And at the metagenesis, basically they reach the inertinite fractions. So we could see sedimentary rocks in any environment with a lot of inertinite in there. And some of them have been sitting there on the surface for millions of years. Like if they would get degraded that quickly, we would not have any organic carbon on the surface of Earth, any rocks, any sediments that has organic carbon. So again, it's a geological perspective that tell us if these bacterial degradation, and I'm pretty sure biology or bacteria has some tricks that we may not know, and I'm pretty sure we will be surprised at some point. There would be some degradation, but in the absence of empirical data, we only have geological perspective to rely on until somebody demonstrate there is such a thing. There has been incubation even up to 8.5 years, and it didn't really release much carbon loss. And one would expect somebody would come out and do acceleration of bacterial degradation just to demonstrate if you increase number of bacteria and food, then you should be able to degrade 50% of biochar. But that hasn't happened. So what do? We use in geological perspective.
Great, thanks, Hamid. I know that topic keeps coming up between the first and second session, but I think all angles that we can touch on that topic is so important and so vital to biochar. We have 2 minutes left now. Harald, I'll turn it over to you if there's any last messages you'd like to give, as well to you, Hamid, with the time we have left.
Yeah, from my side, not necessarily nothing too important. I just found that, and I think you basically said most of what is needed in those 2 sessions. And it's sometimes, I think it's really difficult to differentiate between those topics that we defined previously. But you made it, you did it very well. I think we now have to, we have basically everything together to have the understanding. What I found real interesting when we discussed and had some thoughts on this entire topic is that you showed the carbon cycle, the natural carbon pathway, which you elaborated on in the last session. And then one of our board members saw that basically what happens there within the span of millions of years happens in his machine within a few minutes. I think this is something where someone congratulated you on the analogies, the wonderful ones you made. I think that also is a very interesting one, that there is a shortcut in the carbon cycle, and this is pyrogenic carbons. They are produced through pyrolysis, they happen naturally, and we now have sophisticated machinery which allows us to create something, well, basically alike, even more inert than that. And then in the next session now, we will find out how we can twist our processes based on mainly heat and residence time, of course, feedstock and everything, in order to get to the sweet spot where we produce the maximum amount of inertinite, but still are left with a considerable amount of carbons. I think that's very interesting. So basically, we do a shortcut. We through technical innovations produce a material that's shortcutting the organic carbon cycle within a few minutes, and that it is as inert as the natural material, which I think is very interesting. And we do have the technology on hand, so I guess that's self-spoken. We can act now, and we have to do that. So thanks a lot for this presentation. I really enjoyed it.
Hamed, any last words?
No, thank you very much. Maybe the last word is what we are saying here is not trying to contradict the decade of great work on the permanence of biochar. Please remember, it's a complementary. So we're basically in absence of the time needed for incubations and to see the degradation behavior of the whole material. We are using the geological perspective to give you a complete picture, the two sides of the coin, like we are looking at one side, but now we're missing the other side that the geology can produce for you. I'm pretty sure if you read our paper, now with this presentation, you would have a better understanding of what we are talking about. And I really invite you. It's a very simple language paper, so I hope you read, and if you have any questions after, I'll be happy to answer.
Hamed, thanks again for another great chat. That last point you made of simple language in the paper, I must add some emphasis to, because it really is written in a really clear manner that anyone can pick up and understand. So again, Hamed, thanks again for your time. Next week, we are having a members only discussion, kind of getting back to Haveld's topic of the recipes for optimizing biochar and pyrolyses for permanence and inertinite. So that'll be disclosed only for our members. To our members, please look at your mail feeds. The links will be provided through those channels. And thanks again for everyone joining today. We had about 126 at most participants today, which is a great turnout. So I know we're over time. Thanks again, and have a great week all.
Okay, thank you.
Thanks, everybody. Thanks, Hamed. Bye.
Bye.